Hello students, in this lecture we will be seeing the histone modifications, histone acetylation, histone methylation, DNA methylation and some epigenetic mechanisms that mainly regulate the gene expression. So histone acetylation, that means the acetyl group is transferred into a histone protein. So we know that the genetic material is stored within the nucleus, within the histone proteins. So the histone proteins mainly helps packaging the DNS tightly. So two enzymes are important for histone acetylation. The one enzyme, the HAT, histone acetyl transferase, and the other one is the HTAC, histone deacetylase. So HAT, from the name, it mainly transfer acetyl group, and the HTAC, which is the deacetylase, that means it removes acetyl group from histone. So in this figure, so there is two states of the chromatin or histone proteins, two states in this figure. The upper state is the tightly packaged state or condensed state, and the lower figure in it is the relaxed state or loosely bound state. So why this is happening when the histone proteins are acetylated? So in this you can see the arrow, the histones are acetylated. In this yellow state it is decondensed or loosely state, and when the acetyl group is removed, or when the deacetylation occurs, the histone is tightly packaged. So these two states depending on the modification. So the modification here is the acetylation. So acetylation neutralizes the positive lysine residues. I have talked about that the histones are positively charged and the DNAs are slightly negatively charged, and those interaction, positive negative interaction, mainly holds the genetic material tightly in a very small space. So in this structure, in this if you see that this lysine, mainly the amino acid residue lysine, so this is originates from the histone protein. So this is positively charged, as histones are positively charged proteins. So this ammonium ion, quaternary ammonium ion, this is positively charged, and which is replaced by acetyl group in the presence of histone acetyl transferase enzyme. So this acetyl group is transferred in the lysine residue of the histone protein. So as a result, the histone becomes relaxed or decondensed, and the DNA becomes loosely bound, and then the RNA polymerase and transcription factors can come and do reading, or transcription process can occur. So that means the gene expression can occur. So that's mainly histone acetylation, and we'll be seeing in details in the next slide how it occurs. So don't worry about the figure, it is quite easy to understand. So the three terms: writers, readers, and erasers. So writers mainly add functional groups, in that case the acetyl groups. So writers mainly add acetyl groups to the histone. So in that case the writers are HAT, the enzyme I talked about, the histone acetyl transferase enzyme. This is the writer. And readers, they do not change or do not add anything to the histones, but they helps in reading certain change, or they helps in reading the transcription factor TFs. And so that's the readers. And the erasers, from the name they remove the groups, functional groups or modification. So depending, in that case, in case of acetylation, the erasers is HTAC, so that the enzyme histone deacetylase. So in the earlier slide, if you can just recall, there's the lysine residue. So this was the lysine residue. So in the lysine residue of the histone, the acetylation occurs. So in this figure, so this is one histone or one nucleosome. So in that histone, this is lysine residue. So if you can see, acetyl group is attached in this lysine. So acetyl group is attached, and further attached, So as a result, if you can see, this acetylation occurs and these histones are going far away apart, so loosely bound. On the other hand, if the H tag is in function, then the it removes the acetyl groups. So you can see the acetyl groups are removed, and then as a result, the chromatins or histones become tightly packed, and there is no acetyl group in this. So this is also called repression. That means that suppress gene expression or inhibits gene expression. So this is the mechanism of histone acetylation, and we will be seeing how the histone is methylated, so another modification. So moving to the next slide. So histone methylation, so it is not like histone acetylation. That means the function is not like the acetylation. Histone methylation works slightly differently, because histone methylation can activate gene expression, and it also can suppress or repress gene expression. So depending on the type of methylation, type means not type exactly, the position of the methylation, that which side chain, like lysine or arginine, which amino acid, which histone, and which side chain it is methylated. So the side chain, or the location of the modification, or the location of the methylation, mainly decides the function of gene expression, or the function of the state of the chromatin or histone. So in this figure, you can see the example of how the histone is methylated. So the three terms you remember, if you can remember, the writers, readers, and eraser. Writers add different groups, and in this example, that group is methyl group, and readers just read those changes or modification. They cannot change or add or remove anything. The eraser mainly removes certain groups, for example, methyl groups here. So one thing you have to account for, that is this acronym H3K4. So H3 means histone 3, K means the amino acid uh, label, at least K means lysine. So K is the shortcut of lysine. So 4 is the side chain, 4 number side chain. That means 4 number side chain of histone 3. So there are 4 histones, actually 8 histones, 4 into double, that means histone 2A, 2B, histone 3, and histone 4. So those 4 histones. So depending on the location of the histone and side chain, so this methylation is important. So in that case, if you can see this CH3 is added in that amino group. So this is the writer added the methyl group to a particular side chain. In that case the H3 and K4. So the readers mainly read those three changes. Three methyl groups are added in the side chain of histone H3, and eraser mainly then erase those methyl groups. If you can see this, there is 3 methyl groups, and eraser just remove 1 methyl group. So this mechanism is um, this upper example is activation mechanism. That means this change is activating gene expression. So depending on the location of the histone and the side chain of the histone, this methylation is activating certain genes. On the other hand, if you just follow this lower example of this is same example, but the location is different. The side chain location is different. If you just notice, this is H3K9. So the histone was the same, H3 histone, but the side chain is different. So side chain is line number side chain of the K means the lysine amino acid. So the writers again, they mainly add the CH3 methyl groups. So these methyl groups are added. So if you just follow this my cursor, that here there is no methyl group, but the writers added a methyl group, and then further 3 methyl groups are added, and reader just read these changes, and eraser mainly remove those methyl groups. So in these both cases, the addition of the methyl groups occurs and then reading occurs, and erasers remove those changes, but pathways are different, the location is different. So in the upper example, this changes 3 addition of the methyl group mainly activates the gene expression. On the other hand, this in the lower example, if you can see, this 3 addition is the same addition, but different is the side chain in the location. So these 3 changes, methyl groups, addition of the 3 methyl groups, mainly repress gene expression. So this is quite important. Unlike histone acetylation, histone methylation works differently depending on the location of the histone and location of the side chain of those amino acids. So moving to the next slide. So here is we talked about DNA methylation. So earlier we talked about histone modification. So there was no changes in the DNA. So there was changes in the histone, that means the proteins. But here we are talking about direct modification in the DNA molecule. So DNA, we know that four nucleotides, four different nucleotides mainly make up the DNA, which are adenine, guanine, cytosine, thymine. So in this example, just know that CpG islands, which are mainly CG, if you just recall, CG means cytosine and guanine. So CG are complementary. CG can um, bind together, and other complementary nucleotides are the adenine and thymine, AT. So in the AT there are 2 hydrogen bonds, and the CG pair there is 3 hydrogen bonds. So the CG islands or CpG islands, that means around 70% of the genes. We know that we haven't talked about the transcription process, but just know that for transcription, for it to be occur, there are in the upstream of the location of the gene, in the upstream there are promoter regions. Promoter means at first of the gene expression or transcription, the promoter region mainly activates this transcription initiation factor. So just know that CpG islands are associated with 70% of the gene expressions promoter region. So in these CpG islands, this DNA methylation mainly occurs. So DNA methylation mainly occurs at cytosine nucleotide. So in this figure, this is cytosine, and this methylation occurs. Methyl transferase enzyme mainly transfers this methyl. So this is methyl cytosine, so methylated cytosine. So this is one modification. Mainly methylation occurs at CpG islands, especially on the cytosine. So what happens when the uh, cytosine are methylated? What happens then? It mainly the methylated regions of the CpG island mainly inhibits gene expression. So if some genes are need to be silenced, then those genes promoter regions then gets highly methylated. So that means those genes are not available for transcription initiation start, then there is no gene expression. So in this figure, we know this is the structure organization of the DNA, how it is wrapped. So if there is a gene starting from the right side, then this is the promoter region, and there is a lot of CG, so complementary base pairs. So CG, these are the CpG islands, and in this here you can see this methyl group is added, that means the methylation occurs, and this mainly inhibits this corresponding genes transcription. So I have talked about how it works, decrease or no methylation in active gene expression. So if there is no methylation in CpG islands, those genes will be expressed. But if those CpG islands or these promoter regions are methylated, then methylation inhibits gene expression. So yes, DNA methylation at cytosine inhibits gene expression, and 98% of the DNA methylation occurs at CpG islands. So the DNA methylation, I am talking about the direct modification of the DNA, mainly occurs at CpG islands. So 98% of the DNA methylation occurs at those CpG sequence islands. So more than 50% of the genes. So I talked about 70%, but in some studies 70%, in some studies 50%, more than 50% human genes initiate transcription from CpG nucleotide. So yes, that means gene expression occurs, more than 50% gene expression occurs from the CpG islands, and the promoter region of those gene mainly resides in those CpG islands. And in most types of cancers, we can see there is aberration and abnormalities in those CpG islands. That means those CpG islands get methylated or hypermethylated, and those methylation mainly inhibits those necessary genes. Those genes mainly suppress the tumor. So if those genes are inhibited, those genes mainly suppress the tumor. That means the tumor cannot be suppressed anymore. So as a result, the tumor gets free pass from those suppressor genes, and oncogenes mainly drive those tumors growth. So moving to the next slide. So so as So I talked about those modification, histone modification, DNA methylation, and we also talked about the non-coding RNAs and their function, how those non-coding RNAs mainly regulate gene expression. So these three mechanisms, three different layers of mechanisms that mainly those histone modification and DNA modification and those non-coding RNA, these three mainly falls under the epigenetics or epigenomics. So these mechanisms mainly regulate gene expression, which genes at what point needs to be expressed or not. So in this figure you can see this is the DNA methylation occurs at the DNA level, and here the histone is modified. So depending on the modification, the histone gets active or repressive. That means active means active for gene expression, or repressive means repressed for suppressed for gene expression. The repressive also means heterochromatin. So heterochromatin is the tightly packed state of the chromatin. So if you just can follow my cursor in this slide, in this figure, the acetylation occurs. So acetylation then moves through this phase of the DNA. So acetylation mainly activates the gene expression of gene. Also it activates DNA duplication or replication. So depending on the cell growth or cell division, it mainly DNA replication, or depending on the regulatory phase or the other state of the cell, it mainly works as a uh, gene gene expression or transcription. So when the chromatin is available for, when the DNA is available for transcription or available for its activity, then it can follow two pathways. That is one conventional pathway. We know that it can produce protein. So for it to be for it to produce protein, that means it need to produce a mRNA. So this is one pathway, producing mRNA. The other pathway is producing other RNAs that are non-coding. So this is the third mechanism, the non-coding RNA. So the conventional mechanism from the DNA transcription is the protein production or protein synthesis. That means the production of the mRNA. But the other hand, you can see this transcription process can also generate other RNAs, non-coding RNAs, which mainly further regulates this histone modification or the epigenetic factors, or this further can assist in the conventional um, process. Not assist, but this can also modify the conventional translation process. So non-coding RNAs mainly also regulate this whole process, whole DNA methylation or histone modification. Also non-coding RNAs regulate the conventional protein production system from mRNA. So I guess this mainly sums up everything. I have tried to simplify this histone modification, DNA modification, and the non-coding RNAs, how those are interlinked, how those mainly regulate this gene expression. So this falls under the epigenetic mechanisms.